Hello guys, welcome to the channel. This video is about the alternative pathway of complement activation. In previous video we have discussed about the complement system and the classical pathway of complement activation, which was a part of humoral immune system. In this video we will discuss about the alternative pathway of complement activation. Before diving into the topic, let me give some basic ideas of this topic so that you can easily grasp the concept. Okay, so the alternative pathway of complement activation is a part of innate immune system. As it is a part of innate immune system, it doesn't require any kind of antibodies for its activation. Rather, it get activated in response to other components like foreign surface, okay, that is bacterial cell wall or fungal cell wall, viruses, damaged host cells, or the endotoxins. Endotoxins are the chemicals released by pathogen which activate the alternative pathway of complement system. As it is a part of innate immune system, it is continuously active at a low level by a process called takeover. Its functions are mainly opsonization, inflammation, lysis of pathogen cell wall. Let me discuss about the opsonization and what opsonization means. Let's suppose let's suppose it is a pathogen. Okay, it is a pathogen. Now, this pathogen will be completely surrounded by complement proteins. Okay, and these complement proteins increase the appetite of a macrophage towards this pathogen. As the appetite of the macrophage increase, this will easily engulf the pathogen. This process is known as opsonization. Lysis of pathogen cell wall is a process of killing the pathogen by forming membrane attack complex. We'll discuss about the membrane attack complex later in this video. So now let's dive into the concept. The alternative pathway primarily depends on the C3 protein. Let's suppose the shape of C3 protein is like this. There are many steps by which the complement system is activated by alternative pathway. The first step is to is the spontaneous hydrolysis of C3 protein. The C3 protein it gets hydrolyzed and form C3H2O. Now this C3H2O binds to a factor V and form C3H2O plus factor B. The second step is the formation of C3H2OBB. A factor D a factor D cleaves the factor B into BA and BB. Now the BA will be removed and BB will be attached to the C3. This form a complex known as C3H2OBB. Now this C3H2OBB is called a fluid phase of C3 convertase. This complex sits on the membrane of pathogen by penetrating its membrane and converts C3 molecule into C3A and C3B. After formation of lots of C3A and C3B, all C3B binds to the membrane of the pathogen like this, and all C3A will be released. Now, the pathogen is completely surrounded by C3B protein and it is ready for the opsonization. Here you can see this pathogen is completely surrounded by the complement proteins. These blue are the complement proteins and this is the macrophage. This complement protein increase the appetite of macrophage towards this pathogen and now this macrophage will engulf this pathogen. This is called the opsonization process. Now, if another C3b molecule bound to the surface complex, this will form an efficient C5 convertase. Now, this C5 convertase convert molecule of C5 into C5a and C5b. Now, C5b will be attached to the membrane of the pathogen and C5b after attaching to the membrane of pathogen recruits C6, C7, C8 and C9. These four complement proteins forms the membrane attack complex. After formation of membrane attack complex, they make pores into the membrane of the pathogen and kills the pathogen. This was all about the alternative pathway of complement activation. Thank you guys.